more people will trickle in. So,
okay. Well, we will go ahead and get started then. Welcome everyone. My name is Todd Sweet, and I just have a few Zoom housekeeping items to take care of before I hand it over to our faculty hosts. As you can see by default, we have audio and video turned off just to keep distractions to a minimum and to help everyone's internet connections stay stable. We've also enabled live transcript for those who need closed captions. There's an icon at the bottom of your screen that says live transcript, and if you click that, it will enable closed captions. And we encourage participation, but because we have people muted right now, if you want to ask a question, go ahead and use the chat feature. And that's another icon at the bottom of your screen. And if you click that and just type in your question, addressing it to everyone, we'll be monitoring that throughout the presentation and asking questions as we go along. You can also ask to be unmuted if you prefer to not type, but to ask your question directly, we'll monitor that as well. So with that, I would like to turn it over to Professor Romit Roy Chowdhury, who is our faculty host for the morning.
excellent. Thank you very much, Todd. Welcome everyone to our Saturday Engineering for Everyone seminar series. I wanted to say a few things about this seminar series and then introduce our speaker today. So Saturday Engineering for Everyone started somewhere around 2014 with the idea that several of the very exciting research that's happening in the UIUC campus needs to be communicated to the broader audience of the Urbana-Champaign community. And in fact, physics, the Department of Physics had started this even before 2014. And at some point, ECE, Electrical and Computer Engineering, we decided that we'll copy that good idea and start one for ourselves, where we communicate things that are happening in computer engineering, electrical engineering, computer science, etc. Since then, many faculty have been speaking in this series, and we've had a great amount of enthusiasm from the Urbana-Champaign community to the extent that we would have that in our department building, and there would be a lot of people. But through COVID, things became difficult, but we decided to still have this online on Saturdays. So here we are in spring 2021 doing this over Zoom. We started with our first seminar last Saturday. That was on climate change and on desalination. And today we have this talk on COVID and how UIUC's own campus actually handled COVID and the various techniques and research that went behind it and in the execution of the COVID management. Our next seminar is around space and remote sensing. That the topic of that seminar is, what does the Earth look like from 100,000 miles away, or maybe 1,000,000 miles away? I definitely encourage you to look at the seminar series, the following talks. And the fourth talk is around robots and how robots and why robots should behave like humans, and what is the roadmap of the future in the area of robotics. So these are the 4 seminars for this semester. I definitely encourage you to come and join the seminar and also spread the word to your friends and family. This seminar is for everyone. It should be accessible to from 8-year-olds to 80-year-old, everyone in the spectrum. With that, let me start today's seminar. It's an absolute pleasure to have Dean Rashid Bashir as the speaker for the seminar today. When I mentioned, when I was talking to Rashid about his bio, he said, please keep it short several times. And then when I actually read the whole bio, I realized why I should really keep it short, because it's a very accomplished bio. And Dean Bashir has been a phenomenal researcher and a visionary in the area of bioengineering. So I had to put in some time to figure out what I'm going to select from his bio and tell you about. So here is the short version. Professor Rashid Bashir is currently the Dean of Engineering at the Granger and the Granger Distinguished Chair in Engineering and a Professor of Bioengineering at University of Illinois at Urbana-Champaign. Previously, he was the Executive Associate Dean at the Carle Illinois College of Medicine. In fact, he was one of the founders. He started the College of Medicine at UIUC, and many of you might know that that college is fantastic. It's doing well, and it's a unique program at the intersection of biology, medicine, and engineering. Professor Bashir completed his PhD in electrical engineering from Purdue University in 1992. And then he went on to co-author many papers and very impactful patents in the areas of precision and personalized medicine, as well as in 3D biofabrication of cellular systems. He's been the recipient of numerous awards, including the Faculty Early Career Award, the EMBS Technical Achievement Award, the Pritzker Distinguished Lecture Award, and so on. He's the fellow of IEEE, AIMBE, AAAS, BMES, and there's a long list after that that I've decided to cut down on. He's really a visionary, and he has brought our engineering campus. He's taking it to new levels, and his role and leadership in COVID has been exemplary. So with that, I'll turn it over to Dean Bashir to tell us about point-of-care sensors for detection of pathogens and how COVID was handled in the UIUC campus. Thank you, Professor Bashir.
Well, thank you, Romit. This is really a pleasure, and thank you all for spending your Saturday mornings here with me and with all of us. I really appreciate the invitation, and it's always a pleasure to talk about the research efforts, and especially in this case where our campus has been, as you know, a national and a world model. So, so I will today talk about sort of the background and kind of try to give you a perspective on point-of-care sensors, some of our work, some of the work with our collaborators, and also, of course, mention some of the work from campus briefly. And please stop me at any time for questions. If I don't get through all the slides, that's probably a good thing. So, okay, so let's jump right into it. I'll just start with this slide as just my interest and my passion, and I think a tremendous need around the integration of engineering and medicine. So this is a perspective that I had the pleasure of writing with three other very senior leaders in bioengineering. Professor Bob Narum actually has since then passed away, but Xiu Chen at UCSD and Roderick Pettigrew, who's now actually at Texas A&M, leading their engineering-based College of Medicine effort that's modeled after ours. So I just point this one thing, that we posit that the integration of engineering into medicine and medicine into engineering until boundaries vanish will play a critical role in achieving these broad and specific goals, which is to try to reduce cost of healthcare, increase access, and increase quality. So there's tremendous opportunities, and I certainly welcome everyone to read that perspective. And of course, since then and before that, and I have to say clearly, the ECE department on our campus and in our college is where bioengineering, the Department of Bioengineering emanated from. And I think the leaders from ECE, have been thinking about this integration for a long time. I'd also welcome you to look at this report on convergence. Came out of MIT and other places together, but the sort of thinking about convergence and that word and the future of health. So there is a lot of very interesting opportunities with this integration of engineering, biology, and medicine. Let me just get right to it and try to give you sort of a broad perspective around what happens in the clinical workflow for infection care and management. And some of this is from a review article we had written a couple of years ago that Bobby Reddy, who is actually now co-founder and CEO of a company that we had started, Prenosis Inc. He's leading that now, but he was here with us at UIUC. So what happens today is that if we just look at the events that might happen, there is an onset of infection in an individual that usually, let's say, happens outside the hospital. And that's really, one of the unknowns is that when does that happen and how early can we detect this, right? But it might take a couple of days for the patient to feel abnormal. And then not everyone obviously goes to the hospital or to the urgent care right away. So it might take a few days, but there's this pre-hospital phase, right? Then you get to the emergency department, whether it's urgent care or ER, and might take a few hours, and perhaps a blood culture is ordered, or other body fluid, body fluid samples are taken. And if the condition is severe or the physician feels that it's necessary, then a broad spectrum antibiotics are given, right? Then if the person is admitted, if the patient is admitted, then they're in the hospital. But one thing to mention is that the blood culture, if you try to do a blood culture to look for bacterial pathogens, it could take many days to get that result of confirmation that there is a bacteria in the blood or not, for example. And usually you wait up to 5 days for a negative result. And then there's a sequence of events after that that takes time to identify the pathogen or to treat the person. And hopefully the patient gets better and is discharged. That's what we would want. So there is sort of 4 broad challenges here that I just want to mention. So one is, of course, there is a tremendous need for identifying the pathogen as early as possible. So this is obvious, but this is really still a challenge, right? We want to do it as early as possible. And again, in an ideal situation, you want to censor a test at home. As soon as you start feeling bad at home, you want to be able to use that test to see if you have that pathogen in your body or not, right? One other thing that I think it's really important for even the broader public to know, right, is that for the case of bacterial pathogens like E. coli, and many others, Listeria, many of the foodborne pathogens, other pathogens in our body, there is a culture step that's done. So whenever you go to the hospital and if a blood is drawn and you ask the physician that, hey, when am I going to get the results for the blood culture? They say it could take a couple of days, easy. And what happens is that the gold standard is that you run the blood culture, which means you let the bacteria grow for up to 5 days to confirm that there is no bacteria. And that even, by the way, is just a sample of blood that's taken. So you take about 5 ml, and I'll show that in a later slide again. So there's a sampling challenge too. So it's really still a challenge about how do you really determine whether there is a bacteria in your body or not. And it turns out that some of the bacteria, these bacteria are, they are also living entities. They can be healthy, they can be stressed. So you can have stressed bacteria that do not culture very easily. So there are non-culturable bacteria or bacteria that don't always culture easily. And in those cases, you don't get them to grow even in a blood culture. So that's another challenge. The third thing is that different people respond differently to a pathogen. So, my son, who's he's much healthier. He's a soccer player. His immune system is probably stronger than mine. So, different individuals would respond differently based on their immune system. So I think it's also important to try to profile or get a status of the immune system itself of the person. Not everyone will respond the same way to the same dose of infection. So how do we quantitatively determine that? That's another very important thing, that even at home, it'd be great if you could say, oh, well, how is my immune system doing today, right? And then the last thing is that this is also a very serious problem, by the way, worldwide, is that we are over treating patients with antibiotics, right? So and again, this is not a criticism on the medical profession per se, because really that culture step takes too long. So physicians can't wait, right? They can't wait. If someone is sick or apparently sick, even if their immune system might take care of that pathogen, physicians obviously for good reason don't want to wait. So they are prescribing antibiotics, and I think they are actually, as we all know, we're doing too much of that, and there is a risk of creating strains that are resistant to the antibiotics, right? So this is another important thing, is that how do you prioritize and stratify who do you give antibiotics to? That's a broad goal also. I won't talk about that very much, that we have a big effort in our group on that as it relates to sepsis, because that's really what one of the major conditions is, which is the outcome of infection gone bad. But I'll talk about the first 3 here, and especially as it pertains to COVID and other pathogens. Okay? So those are 4 broad goals that I think there's a lot of opportunity to make an impact. Okay? So let me also show, this is a busy slide, but just to kind of get a perspective, in an ideal world, this is what you would like to do, right? you would like to be able to take this sample from the body if someone is infected, and the source can be a variety of sources here, from whether it's saliva, sputum, urine, venous blood, or a finger prick, right? Depending on the virus, depending on the bacteria, where it's found most, depending on the condition, you take a sample, and then you want to be able to do rapid detection. So this is, I mentioned this already, very quickly detect what's there. If you can isolate that from the person, then what happens in the clinic, some clinical labs, you actually can try different drugs to see which drug works on that bacteria that was isolated from the pathogen, right? Doesn't always happen, but this is certainly a goal, right? At the same time, right, at this other pathway, at the same time, if I can take again the patient's blood and look at the profiling of the immune system, how is the immune system doing by looking at different cells, different proteins, different DNA markers in the blood. And based on all that information, I'd like to be able to then design the drug/therapy management regimen for that person and administer that back, right? You want this process to be as fast as possible, right? And of course, in today's day and age of data analytics and machine learning, what we would also like is to collect all this data and be able to learn from that, see patterns, so that you build a big database and then of thousands of patients. And when the next person walks in to the hospital or to the urgent care, you want to test them and be able to compare them with the thousands of patients and be able to maybe even project what might happen. So that's sort of another high-level goal for what we call personalized medicine for infections.
Ask a quick question.
Please, yes.
I'm coming from my deep knowledge in biology. Let me preface my question with that. Okay, super naive question. When you draw a sample of blood,
yes,
is it guaranteed that the things that I'm looking for are going to be present in that sample? I mean, is it possible that my bacterial infection is somewhere in my kidney or somewhere else? How should I think about that, right? Is that sampling always going to be guaranteed?
That's an excellent question, and it's a real practical question. It's not that's a real problem. That is a problem. I think the goal of detection of a pathogen is like a needle in a haystack. It's a very similar problem as trying to detect a cancer tumor really early on somewhere in the body, right? So that's right. Pathogen that respiratory pathogens would be in the respiratory tract, right? Or the lungs or the saliva or the nasal cavity. Pathogens that have now entered the blood could potentially be found in the blood, but there are other pathogens and other organs that might not have yet moved over or gotten into the blood. So that is a real challenge. As a matter of fact, what also happens in many cases, for example, is if there are within your digestive tract, as you know, there's a lot of bacteria. If there is any injury or insult to the system there, those bacteria can cross over into on the blood side versus the other side. So that's a really good question, and that's a real challenge.
And a quick follow-up, if the bacteria is in the blood,
yes,
then can we be sure that 5 milliliter will capture it?
Again, a very good question. So that's the current clinical standard is to collect roughly between 5 to 10 mLs. And the sense, I mean, that number also comes from a clinical practice with the idea that, well, if you run, enough of those every so often, you will be able to at least catch what's there. But that's exactly the problem and the challenge, is that sampling is just that exactly. It's just 5 ml of blood, right? So it is a small percent of all the volume of blood that I have in my body. Now, blood is circulating, so you have a good chance that you get that you will capture that, but that is not enough for the sampling. And that's one of the challenges in this space, is that you also cannot draw a lot of blood, right? Just because of that could cause issues with the patient. That sampling, for example, so for kids and infants, half an ml to 1 ml is taken, a lot of times from the toe, right? So that's a real challenge, is that how do you identify this foreign, non-human entity within the body and within the blood? Yeah. And in an ideal world, you want to be able to take enough samples often enough to be able to do that, or figure out some in vivo measurement also. Somehow continuously measure as blood is flowing, a sensor that could look inside the body through the skin, for example, and be able to measure that. Okay, great question. So let me also just, again, for our information's sake and knowledge is probably more important, but let me just, so there is, what happens in the current labs, right? So you could be looking for bacteria. And in that case, this is kind of what happens. 5 ml of blood or urine, you culture up to 5 days. And then what happens is these bacteria, most of the ones that are found in the body from a disease perspective, they double every 20 to 40 minutes. Okay, so that's why when you culture them, they double every, so in a few days, you could have up to 10 to the 8 to 10 to the 9, right? 1 billion bacteria, let's say, right? So then when I take a small volume of that culture solution, I'm guaranteed to have enough bacteria in it. So if I just take 25 microliter after a couple of days, I'll have thousands of bacteria in that small volume. And hence, then I can take that small volume and run it through a PCR machine, and I'll talk about that a little bit, a PCR machine, for example, right? And identify that bacteria. So this is why for the culture, we always talk about the sensitivity being down to a single pathogen in the volume you are collecting, okay? But it's because of the fact that you can amplify and that bacteria can double every 20 to 40 minutes. So if you wait long enough, you're gonna have more than enough to get in a small volume, okay? That's really important, I think, to kind of get. Of course, then there are fungi, yeasts, and molds. Believe it or not, they take even longer to culture. Their growth is even slower. So that's, you would never wait for that. But it does happen in some cases to identify a particular unusual pathogen in a body. These cultures are done in clinical labs, but they can take 2 to 4 weeks. They're even slow growing. And then you have a similar process there, okay? Now, viruses are technically not living. Viruses need a host. Viruses by itself cannot amplify or grow. They have the RNA or the DNA and then the envelope to infect, but they have to infect a cell host. So viruses do not amplify outside a cell line or outside the body. Okay, you have to actually, there's no easy way to grow them in the clinic. So what you do is the limit of detection for viruses is actually a lot higher, essentially. So if you were to draw 5 ml of blood or urine, the fact is, in this case, a sensitivity is having enough particles in the volume I'm testing. So which means that it's about thousands of particles, as they're called plaque forming units. Let's say 5,000 PFU in 5 ml would correspond to 1 PFU in 1 ml. And hence now, if I take, let's say 25 microliter, I'm gonna have at least 25 particles in it. So this is really important to also understand, is that the fundamental limit, sensitivity is a lot more.
Rashid, I think we lost you.
In the clinical labs that measure different types of cells and cell counts. And then there's also something that's the flow cytometer, which can measure cell surface markers and other information that's important for diagnostics. Okay, so I wanted to kind of just give this background of the limitations of and the sensitivities. Now, let me also, for also this is important to understand. We hear PCR a lot in the general, on our campus. We all are aware of our saliva test, which is a PCR test. So nucleic acid, there's 2 types of nucleic acids. There's, well, two major types. There's more, but there's RNA and DNA. And you can have pathogens that can be either RNA pathogens or DNA pathogens. If it's an RNA, like a virus, like SARS-CoV-2, you have to first convert that RNA to a DNA. And there's a step to do that. The molecular biologists over the years have figured that out. It's called an RT step. So I just, the fact is you can convert an RNA to a DNA if you start with an RNA. If you start with a DNA, then you don't need that step. And then there is this, of course, the gold standard is called the PCR, polymerase chain reaction. Whenever you are using PCR for an RNA pathogen, you add this RT. So you might hear RT-PCR if you really hear the technical jargon. This is one of the gold standards. And at the end of the day, this is, again, an amplification-based step. So there is multiple temperature cycles that are run. Takes about 90 minutes to run this test, let's say, in a commercial system. And the sensitivity is those few copies per the volume that I am analyzing, okay? There is also another step, another type of amplification, which what we use a lot in our lab, and there's some commercial examples that is called LAMP, which is loop-mediated isothermal amplification, and that's another amplification process, method that does not need the temperature cycling. So hence the instrumentation can be smaller and it's easier because you just keep the solution at one temperature. Okay, so that's kind of what you want to just broadly remember. Those are the two most commonly used amplification processes for DNA or RNA testing. Okay, let's talk about SARS-CoV-2. So what to test? And I think we all have learned about this more than we wanted to, but so this is kind of a busy schematic, but just the left is showing, this is writing a review article on this. So this is an image that was drawn. So you have the sample sites, right? You have the nasopharyngeal or nasal swab, or you might have the saliva, which we believe is a really good source. And of course, there could be venous, blood that's drawn, but you're testing for different things. But the three types of tests that you hear about. One is the molecular test shown here, kind of on the top here. These are the RNA tests, right? So molecular test is the most sensitive, the gold standard. And there you are testing for the RNA of the virus. In this case, the target is amplified. This is a very sensitive test. It's the gold standard. This cost is probably, I'm putting on the very low end. It's more like, 15 would be the very, very, very bare bone cost of the reagents and everything. It's more than that. It's maybe $15 to $20 to $25, depending on the infrastructure. If you run this test in a clinical lab, the reimbursement cost is over $100. Could take 30 to 90 minutes, more like on the 90-minute side. And this doesn't include the time to report the data back, right? That's important if you're doing this at scale. A second test you hear about is this shown in the middle here, which is the antigen test, right? So antigen test, again, just to be clear, right? It is less sensitive than the molecular test. And it is because the target is not amplified. So in an antigen test, I am looking for proteins or fragments from the virus. It is not looking for the RNA. And proteins do not amplify. There's no process such as amplification of proteins like it is for amplification of RNA or DNA. So target is not amplified. Hence, it is inherently about 100 times less sensitive. So antigen tests are less sensitive by default, the commercial ones at least. And here, some of our colleagues like Brian Cunningham and others are leading the way in developing very sensitive future new antigen tests also. But everything that's available in the market today, it is about 100 times less sensitive than the PCR tests. These tests can be cheaper. They are more available at the point of care, and the results are faster. It takes about 5 to 10 minutes to get the results from those tests. And then the third test you hear about is the antibody test, which is from the blood, which is whether the person has the antibodies against SARS-CoV-2, which means either they have the vaccine or they've been infected. This test, again, is very similar actually in how it's run with the antigen test. So there's sufficient sensitivities because you develop enough antibodies in the blood. But again, target is not amplified. And this is very similar. The technology that's used is very similar to the antigen tests. So I didn't want to kind of mention these three. And then over the year, there's been some, sometimes there's confusion across these three, but these are three specific different things to be testing. All right. Just briefly, the urgent care, I mean, the bottom line is that these, the antigen tests are available at the urgent care, but the molecular tests are not still. So tests in the urgent care are the rapid antigen tests, or you take a sample and you send them to the lab for the molecular test, okay? COVID-19, I think we all know, is expected to become more endemic, like a flu, essentially. So we're all, we believe that in the future, there'll be a flu test, and then you also will be testing for SARS-CoV-2 for COVID-19. So the current number of tests that's run clearly is expected to decrease for COVID-19, right? So it might be 100 to 125 million tests in 2021, 30% of them being molecular tests. Again, these are projection based on this year, and this number is hopefully continuing to increase. But the fact is that over time, the anticipation is that COVID-19 testing would probably be similar to like a flu test, where you get it done, probably more often than flu initially, but then eventually just like flu. Just as an FYI, in the US, right, there's about 40 million rapid flu tests performed, about 25 million rapid strep tests performed, and about 4 to 5 million of STI-related, sexually transmitted disease-related, infections-related tests performed. So that's another opportunity, is to provide the molecular test at the urgent care, which today doesn't exist. Okay. Again, please stop me if there's any questions there. Of course, I will be remiss if I don't mention our campuses test. This has been so wonderful. We're so proud of everything that our campus has done. This saliva test that's been developed from saliva using PCR. So this test, of course, got the FDA approval just within the last month or so also. So this has been, I just want to mention, of course, it's a tremendous effort. We're so proud of everything that's being done on campus. And again, I will be remiss if I don't show this slide on the COVID-19 SHIELD program. Marty Burke is the team lead for this, but lots and lots of people have contributed for algorithms for testing, for the developing the saliva-based test, and then of course the app also, that again, lots of faculty from engineering and beyond have really come together as a campus. And we have the campus effort, we have the SHIELD Illinois, and we have SHIELD T3, which is more across the state. And of course, as you know, as of just a week or so ago, week and a half ago, we're also deploying the test to the community. So this has been, we're just so proud of everything that our campus has really done. Let me also mention about this idea of, again, where to test in a very broad sense, right? So this shows sort of all the possibilities that could exist in terms of the test, and going from left to right, I'm going, as you go from left to right, the size of the lab, call it, becomes smaller and smaller. But as the size of the lab becomes smaller, the fact is that the number of tests also become less per unit time, right? So on the very left, you have the actual lab. So this is what our vet diagnostic lab has been turned into a human clinical diagnostic lab. This clearly, you can do tens of thousands of tests per day in a proper lab, right, where the samples are going to the lab. The lab can be put in a mobile trailer, and I'll give you an example of that again our college and our campus has done. Can do them thousands per day. You can imagine a scenario which is for a benchtop system, where you move the system to an airport or a stadium, where maybe you can do hundreds per hour. You can imagine a suitcase type of a form factor, which we can do tens per hour. And then you can eventually imagine a handheld portable, which might just be able to do a few, 1 to 3 per hour, for example, let's say. And the goal is to make this even faster, by the way, time-wise. But this kind of gives a broad perspective of what the need is. And clearly, this year, everything that's happened this past year since the pandemic, there's more and more examples of these things towards the right, essentially, than ever before. Right? So I want to mention this also. This is something that really proud Abigail Woolridge led this team from ISC faculty that we charged her about 8-9 months ago. To say, hey, can we take what was being done in the VDL in the lab, and can you turn that into a trailer? So she worked with a team, campus-wide team, again with NCSA, with engineering, with working with the vet diagnostics lab, and turned essentially that lab into a trailer, right? And this is what SHIELD T3 then is basically deploying around the country. This is in one of the trucks that's in the Bay Area in collaboration with Bloom Energy. And certainly there is a need for this sort of modality that I think is gonna grow. All right. Now in the next, let me talk about some of the things that we're doing and where we come in. And a lot of our work, of course, is motivated by this integration of micro, nano, and bio. On the right, sort of what silicon fabrication has been able to do in terms of miniaturization. In the middle is what sort of bottom-up chemical self-assembly can do, including 3D printing. And then on the very left, there is just a size scale for various biological entities, including the size of a virus, which is about 100 nanometer or less. Bacteria are a few micron in size, and of course, then you go up the scale. So this intersection of engineering, biology, and medicine is what our passion is. And they have many leaders across the college and campus in this space working together. So one of the things that I'm very interested in is trying to see how we can put some of those testing protocols and those tests on a chip or on a cartridge-sized device, right? So a lab on a chip, where the patient doesn't need to go to the lab, but rather the lab comes to the patient, right? And how do we put different steps that are needed into that cartridge where it makes sense and where it can be done? So we've actually been working on this for many years. Some of the work was in collaboration with Professor Brian Cunningham. I'll show you one example where we'd actually demonstrated, even pre-COVID, that you can do this on a cartridge with HIV virus, HCV, Zika, dengue, and chikungunya, and we had published on this. Many of these viruses, actually most of this, are found in the blood. So the idea was to take a small drop of blood, be able to introduce that in a cartridge, and this cartridge will be the size of a credit card, and it could do that isothermal amplification that I mentioned earlier on this cartridge. And then you could use a cell phone camera to detect the reaction as it's happening and get the result. That's kind of the cell phone-based detection with a small portable reader and a cartridge which takes a sample. So we've been working on this, and so that's when the sequence was released for SARS-CoV-2 on January 11th, actually exactly to be over in 2020, many groups, including ours, as soon as the sequence came out, we took that sequence and we started to design the primers, which are the short molecules that detect specific segments of the genome. And there are these different gene N, gene S, and ORF1A. But the point being that we designed our own primers. There is a methodology to do it where you try to look for the genome, the most conserved regions, and try to identify the sequence. And based on that, you can design what's called the primers, which are the molecules that detect specific segments of the genome. So that process can actually be done pretty fast. Multiple groups did that. We also did that. The work wasn't published till later in the summer, early fall, just because of the reviews and everything else. But we were able to also do that and design the sets of primers that would detect, let's say, this gene N, which is the most commonly detected gene for SARS-CoV-2 testing. And then, again, working with collaborators here with Professor Bill King and Brian Cunningham. Bill King, as you know from mechanical science engineering, he is the world's expert in additive manufacturing. So we designed these cartridges, which in the past we had made with silicon and in glass, but in this case, you can make them with plastics that are biocompatible and do it very quickly. The design cycles are very quick in this case because you can additively print these, test them, and if something doesn't work, go back and change the design. Unlike injection molded cartridges, which take much longer, and even silicon-based devices or any of the processes that were used would take longer than this additive manufacturing cartridges. So you see here a cartridge that's, again, the scale is here, but this is the size of a credit card. We can inject different solutions in it here. The patient sample can be injected here in this port. The LAMP reagents can be injected here. They get mixed in this microfluidic mixer, and then they end up in this region right here, this flower-shaped sort of 6 chambers where the reaction takes place. We put this cartridge in this device, which has a heater. And in this case, it's 1 temperature, because remember, that was the advantage of using this lamp process versus the PCR process. And the cell phone camera, can detect the amplification, and the light intensity increases as the reaction continues when you have a positive. So working then with our colleagues at OSF and Carl, they've been tremendously supportive. We were able to get IRBs approved, both with OSF and Carl. So Dr. De Ramirez is at OSF. She helped us get viral transport media samples from patients from OSF. And then Dr. Karen White, Dr. James Kumar, and Mark Johnson here at Carl, they helped us with getting IRB Carl for VTM and for saliva. As a matter of fact, Carl was tremendously helpful, of course. I hope you all know, but in terms of getting our first clinical study that was done back in May timeframe, where we collected, myself and Marty Burke were the PIs of that IRB, where we collected the first 9 samples from the drive-through from Carl over at Madison Kirby, just south of Kirby, that drive-through, and that was the first 9 samples that were tested with saliva to develop that initial test for the saliva to PCR. So anyway, long story short, we then later also then did this work where you would collect the VTM in this case first. We did this with VTM, where the swab is put in a solution. The viruses, the assumption here is that the viruses are dislodging from the swab into the solution, and then that solution is used to either do the standard clinical pathway at the bottom, which is the RT-PCR, or the top, which is our RT-LAMP process. And that worked pretty well with this clinical study we did and was published in PNAS. Shows that in about 20 to 30 minutes here, this bottom left curve shows that this is a different time, positive and negatives. You have some background fluorescence always, which stays there even for the negatives, but then you can see when you have a positive, that would increase over time, the fluorescence. And these are real clinical samples, and you can detect this increase in fluorescence here shown on the right. This is the images that you get from the cell phone camera. So this is pretty interesting and exciting, and the fact that one can develop potentially tests like that that can be used using VTM, which is the viral transport media from a nasopharyngeal or a nasal swab. We then further did another study where we compared then the saliva with the VTM, and this paper was on arXiv last fall. We put it on, and it's going to the final review now to be published. But here again, we were able to show what you want to see is that this is the VTM and saliva from the same patient, and we can get very similar results essentially with an optimized RT-LAMP on the left of both these sets of curves, and the right side is a conventional 1-step RT-LAMP. But the point to make is that the VTM and saliva in this particular case also are quite equivalent. So we believe that saliva is easier to collect compared to the nasopharyngeal swab, which is sticking the swab, all the way up to deep into the cavity. Of course, as you all know, if any of you have done the Carl test recently, or even in the last couple of months, that they have also now shifted, the clinical practice has now shifted from the nasopharyngeal swab to just a nasal swab that you can self-administer. So the clinical practice is shifting towards the nasal swab, which is easier to do, and one can do it themselves also. Okay, so maybe I'll stop there and see if there's a good, if there's any questions on this part from anyone.
Rashid, I have a quick question.
Yes.
Can you speak to what research, or even engineering wise, what happened that made Illinois the pioneers of the saliva testing that the rest of the country couldn't do? Is it the speed of it? Was it an insight in chemical engineering, molecular engineering? Where was the insight that made Illinois that differentiated Illinois from the other research that was happening?
So that's a great question. I think overall, clearly, the speed and the rapidity at which we did the test development from saliva, having the partnership with Carl to have the saliva samples available, and then being able to develop that test. I think all that was a critical piece of it. There's no question. But then I have to say that I think the reason the whole effort has worked so well is really a systems engineering approach. I mean, the whole approach of the systems, thinking of the whole system, right? And again, our whole campus coming together, thanks to the chancellor and provost, and we're just going to go do this. So I think it was more. It's equally important. Of course, equally important was to get the basic components done, right? I mean, the modeling part that was done so well, so quickly, the testing, the test development, the app that was developed, right, based on the RockWire and other work that we built on really quickly. So those components were critical, but then putting it all together and saying that, okay, let's set up these 19 sites across the campus, collect the samples, run them over to the lab, right? Set up the 3 shifts. So I think it was that willingness to do the whole organizational effort to put in place. I mean, and I think it was really a tremendous leadership from that perspective. And that's why, as you all know, right, in fall, there was parts of fall that 2% of the national tests were being done here on campus. I mean, there was no fundamental reason why the testing couldn't have been scaled up in other states or nationally, right? It was a matter of really the fact is the leadership and saying, we're just going to go do this. The components are developed also. We're going to put it together and make the whole system work. Because the test itself is really important, but a component. You have to figure out how to get the sample to the lab really efficiently, right? You have to figure out how to get the results back to the patient. So I think it was a combination of all those things come together really well.
So is it fair to say that the engineering and the chemistry and the molecular underpins of this work, they were known, but it's bringing them together, which was the important part?
So what was not known, right, is the fact that will the saliva test work? There, at that time when we did it, just about that same time, right, there was a report from Berkeley, and there was a report from Yale and Rutgers. So there was studies were just about to come out as we were doing our study too from saliva. So I think that was, Marty, Paul Horgan-Rother and their lab, and Tim Fan, and I mean, trying to make that decision from a clinical perspective too, that we believe that saliva would be equivalent or better, we believe in many cases, than the swab. And then just kind of making the test work. That was very early knowledge, is that would you find the viruses in saliva, or would you even find viruses in saliva that are not in the VTM? So I think there was that unknown, but it was validated pretty quickly, because I think this was also a great example of how academia, we don't always work on a problem that's urgent. We're just trained to work on something that's long-term. Right. And truly, if you stop everything and you say, here's a problem, let's put our talent towards it, I mean, we can solve that. So in a way, it's been a good example of, hey, we should really solve some other urgent problems like this and not wait for a crisis to happen.
So if you think of literature, there is not much work on trying to detect viruses from salivas?
There was not very much work. There was some work before that, but SARS-CoV-2, obviously, there was not very many reports. And even still, there's not too many reports. There's a handful of reports on that. And the earliest test, was the one from Rutgers and Yale and ours, and a report from Berkeley.
Wow, thanks.
Okay, so I think in the remainder of the time, I know we're approaching, I'll just maybe try to skip through actually these 2 points, but I just want to mention the other 2 challenges I do want us to remember at a high level. The blood culture problem, right? If anyone can find a solution to that, by the way, a tremendous clinical impact and tremendous commercial impact. This is a huge opportunity for startups and for companies. Everyone is looking for a rapid way to detect pathogens, which eliminates the blood culture. So I'll just show, we do have an approach here that we have early demonstration, which is inspired, by the way, this idea of a dried blood spot technology, which is already used where you put a drop of blood on a filter paper. It's essentially like a filter paper. And it's been shown then to preserve the DNA and potentially the RNA in that dried format. And then you can ship this even to the labs, and they would elute this out and then run the PCR. So this was really only been demonstrated and in practice with very small volumes. So we were kind of inspired by that. We said, wait, what about, well, why don't we do this with large volume? If you can imagine a large volume that you could somehow dry. So long story short, we came up with a process where you can actually heat and dry the blood. And this here on the right is what dried blood looks like. So if you take blood and literally heat it and dry all the liquid in it, the solid will look like this on the right. These are scanning electron micrographs of dried blood, which has the solids, the blood cells, and other matrix. And then we can do a reaction in these solid liquid phase, what we call a biphasic reaction. So we can actually show that you can detect E. coli directly from almost an ml of blood in 20 to 30 minutes, and the whole process takes 2 hours. So we're pretty excited about this technology. It's still early stage, but we're kind of moving that forward to try to eliminate blood culture and directly get results of few pathogens from whole blood. And then the last thing, let me skip this. This one is kind of might be interesting. A more recent result on using graphene. So graphene is, people have heard about graphene. It's a monolayer of single layer of carbon atoms. And the fact is that because it's so thin, it can actually bend. So as a matter of fact, in our college, we have a NSF center. It's a material research center that Nadia Mason is a PI. She directs the center. I'm also a part of it. And the idea here is that when you bend graphene, what other interesting properties can be manifested? And it turns out one thing that happens when you bend graphene is that if you bend graphene and then put it in a liquid, you have some very interesting charge properties that happen at this interface because of these nanoscale curvature. So long story short, we made transistors from that, and we show that you can detect molecules at a very, very sensitive level down to what's, attomolar range, which is just, tens to hundreds of molecules in the sample. And most recently, we also showed that you can use that bent graphene sensor, or crumpled graphene sensor, as we call it, to actually detect the COVID, S protein and and the spike protein and the N protein. So you can actually detect the COVID protein against a background using this graphene sensor. So this could be an interesting possibility to have a very sensitive antigen test that has a sensitivity similar to the molecular test, very similar to the molecular test. But it's clearly much earlier stage and more work needs to be done to do clinical studies. We did these control measurements with this, and they seem to work really, really well with these curved graphene samples. Okay, with that, I'll just show my last slide. And this is actually a slide that I had made up to present at a conference that NSF organized a couple of months ago on predicting pandemic. So it's like, what would you want to do for the future? What have we learned? So, I mean, this is just some thoughts where, we know, well, we hope they won't, but most likely the likelihood of more pandemics in the future is there, right? So NSF had put this workshop together with a very, very multidisciplinary team of faculty from all across to say, well, what could we do in this space to predict emerging pandemics? So I thought that, well, this is an approach, maybe again, it's a systems approach, perhaps. I mean, I put the word engineering, but really systems approach. So, you want to think about monitoring the pre-indicators of pandemic, right? So there could be environmental, epidemiological, maybe population and illness data from social media, food and water supply. And then, the next thing clearly is to be able to collect and sequence genomes of all pathogens, right? So this is, of course, a very costly effort, but there are some efforts like that that are launched if you do a search on this from in the UK and some in the US. But perhaps you can narrow those pathogens by saying, well, what are the highest risks, right? So I think the biologists and epidemiologists and genomic experts can predict what are the top 15 pathogens that have a potential to cross over and get to humans, for example. So thinking about the highest risk, but also current and future. So as soon as there's a break, there's an outbreak, you want to be able to sequence that, because then you can develop the test very quickly, right? Then you also need to figure out, well, what, where, and how often to measure. So if you define a pandemic as something that crosses national boundaries, there's actually a definition of, CDC has a definition of a pandemic, and so has WHO has a very specific definition of, which is starts in one country and then spreads out. So you want to be testing at airports, train stations, water, food, environmental samples, of course, human samples, but you can't test everybody in the world yet. So the question is, how do you stratify, how do you measure who to test? I mean, how do you decide who to test? And then, of course, mitigation strategy, public health, and the policy part of it is really, really important, right, in terms of getting adoption, but also putting some policies in place. And then once you have the sequencing and all this information, clearly drug development, which I think has been such a heroic effort also this past year, how quickly these drugs were developed. Diagnostic scale-up, I mean, we did not do very well, I would say, nationally and as a world in that area, in the diagnostic scale-up, we didn't do very well. I'm proud to say that our town and our campus did really well. And then, of course, deployment and distribution. So this might be some thoughts around future, pandemic. So with that, I'll stop, and I'll thank, of course, my research group over the years, tremendously bright students and postdocs and staff. And of course, we collaborate with lots of people that I had here on a slide over the years on various projects from our campus and beyond. But with that, I'll stop and say thank you.
Thank you very much, Rashid. That was very exciting and very inspiring. Questions? Okay, let me start, and maybe others will follow. Rashid, so broad question: if I gave you a magic wand and said you can solve one problem with this wand, and that problem, which one would you solve to make the biggest impact? So in this whole sequence, in this whole systems, where there are multiple modules that are interdependent, do you have a sense of this one or these two, if we can solve from an engineering perspective, the return of investment is the biggest?
That's a tough question. There was one thing. I mean, I think from a, I would say from a, okay, so there are critical steps that are needed to do other things, right? And I think getting the sequence of the target pathogen is critical. I mean, if you don't have the sequence, which means you have to be able to isolate it. If you don't have it isolated or have the sequence, then you can't develop drugs, you can't develop diagnostic tests. So that's one really fundamental thing. You have to be able to isolate and sequence. Then there is a whole bunch of other important engineering, policy, implementation, manufacturing, and FDA approval related things, right? Things that take many years to approve. This year, because of the urgency of things, this EUA approval mechanism was used to move multiple point of care technologies, by the way, that were in the market or were being developed before. They got approved much faster this year than they would have ever done it, right? So there is, I think this, the fact that, crisis is a mother of invention or translation, I would say, in this case, that's less of a fundamental problem. It's more of a policy and implementation problem. So I think, yeah, that's where I'll stop. I think if you have the sequence, you can do lots of important things right after that. Scientifically, the challenge is more on the biological end of things about how do you predict really early on when things have happened, right? So how do you set that system in place? That's a very important, I think, challenge. And again, that system, I believe, can be put in place, but it's just that, when you don't have a problem, you don't put that effort always, right? So, I mean, the ventilators was another such example, right? Emergency ventilators are things you don't want to have, but when you need it, you better have it. So how do you build up a supply of emergency ventilators, which our country was short on when we needed it? And it took a little long. It took a couple of months from many companies, including the one example from our campus, which was done really fast, led by Bill King. And we even got an EUA approval of that, working with FIT and our partner. But the fact is that when you really need it, that's when you want to have it ready. So thinking ahead and planning and putting things in place when you don't have a problem is critical. Right?
Great. Other questions?
There's a question here: how many testing labs are on campus, and where are they? So there is actually one lab at this point, which is a part of the veterinary diagnostic lab. And again, thanks to the campus and the vet school for making that happen. But that's the one lab on campus where all the samples from the multiple sites that the saliva is collected, they are run over to that vet med building in the vet diagnostic lab, and that's where the testing is done. There's 3 shifts running, 8 hours per shift around the clock. The people are doing the testing there.
I have a question about you're asking where to sample in a society, a train station or whatever. You would need to have some kind of model of the whole population of how people flow around and mix. That would be an incredible,
that's right,
detailed model of social interaction.
That's right. That's right. And I know that Sergey Maslov and of course Nigel and their team have been thinking about that. They've done some of this across the state, but exactly, yeah. How do you monitor that flow of people and goods, right? So as a matter of fact, there's some interesting synergies there or learnings from one of our faculty in civil engineering had modeled the food supply movement across the country. And so that's right, exactly. How do you measure and model people flow, people movement? So movement of people and goods potentially, or things that can carry the contamination. I mean, this particular one, right, is carried by individuals and can be airborne. There are others that are only with physical contacts. Understanding that for what pathogen, and then how do you model it across a large body of people is a really interesting problem.
Rashid, my 10-year-old wants to ask, why is it that the second dose of the vaccine make things harder, or people are falling more sick and feeling the consequences or feeling worse compared to the first dose? It seems like the first dose should get you ready for the second dose and you should do better. Any thoughts on that?
Well, I have to say I'm not that's not my area of expertise, so I'll refrain from making detailed statements there. But the general sense is that for the Moderna, right, for example, for that there's two doses and they work in complement, and then the full sort of mRNA. These are mRNA drugs, which again are the cells are programmed to then express the proteins, and it's that's complementary of the 2 doses that work together, and the second one is kind of completes that step, so to speak, to have that. So I'll kind of leave it at that.
Sure.
But the good news there is that certainly more vaccines are being available too and being approved. There's been at least 2 that are single dose also.
If there are no other questions, then let's thank the speaker once again. Thank you very much, Dean Bashir, for this wonderful talk. And thanks everyone for joining in. Again, please tell your friends and family to join in for our next seminar in one of the coming Saturdays. It's not the next Saturday, right, Todd? It's not the next one. It's a Saturday after that.
It's in late April, and I could get the exact date, but not for a couple weeks.
That's right. And I think the list of seminars, I think, Todd, you've pasted a link where all the seminars have been posted. So please look at that link. You can Google Saturday Engineering for Everyone, UIUC, and I think that'll pop up as the first link. So with that, thank you very much, Rashid, for taking the time on a Saturday morning. Thanks everyone for joining in, and I'll see you in the next seminar late April. Bye.
Bye. Thank you, Rashid.
Thank you. Thanks, Bruce. Thanks, Roman. Bye. Thanks, Todd.